Inside every single one of your cells, right this very second, a microscopic process of just staggering complexity is going on. Your DNA, which is basically the 3 billion letter instruction manual for you, is being copied. And it's happening with incredible speed and precision. This is the story of how life makes a copy of itself.
So let's dive right in. We're going to start with the big puzzle. How does DNA even copy itself? Then we'll look at 3 competing ideas that were floating around and the brilliant experiment that finally gave us the answer. After that, we'll meet the molecular machinery, the little enzymes that do all the work, and get into the nitty-gritty of how it's all done, including a really clever solution to what seems like a one-way street problem. You see, back in 1953, when Watson and Crick showed the world their double helix model, they solved a huge mystery and immediately created a brand new one. They could see just from the structure that it had to have a copying mechanism. But how? How did it actually work? That question became one of the hottest topics in all of biology. And this right here really captures the energy of it all. DNA isn't some dusty blueprint just sitting in a library. No, it's an active, bustling molecular factory. This copying process, which we call replication, is fundamental to, well, everything, from healing a paper cut to the growth of a whole new life. I mean, it's one thing to have the blueprint, right? But it's something else entirely to figure out how the photocopier works. How does this elegant little twisted ladder unwind itself and somehow produce 2 perfect identical copies from just one original? The entire scientific community was buzzing with ideas. So before anyone had any real proof, there was this fierce debate. 3 main models popped up, and each one had a totally different idea about how DNA could be duplicated. Let's make the contenders in what's basically a biological detective story.
Okay, so here are 3 suspects. On the left, we've got the semi-conservative model. The idea here is the original molecule splits in half, and each half gets a new partner. In the middle, the conservative model suggests the original stays totally whole, kind of like a photocopier, and just spits out a brand new copy. And then on the right, you have the dispersive model. This one's a bit chaotic. The original gets chopped up and just scattered throughout the 2 new molecules. So which one was it?
Let's take a closer look at our first suspect. The semi-conservative model is just beautifully simple. The 2 original or parent strands unwind, and each one acts as a template to build a new matching strand. What you end up with is 2 DNA molecules, but each one is a hybrid, half old, half new. Now for suspect number 2, the conservative model. This one imagines the parent DNA molecules staying completely together after it's copied. It acts as a template, sure, but then the 2 original strands snap back together, leaving a completely new double helix to form on its own. So you'd get 1 old molecule and 1 totally brand new one. And our final suspect is the dispersive model. This is definitely the most jumbled of the 3. It proposes that the parent molecule gets busted up into little segments. These segments get copied, and then the whole thing is reassembled into 2 new DNA molecules that are basically a mishmash, a mosaic of old and new pieces all mixed together on both strands. With 3 competing theories on the table, the stage was set for one of the most elegant and frankly beautiful experiments in the history of biology. In 1958, 2 scientists, Matthew Meselson and Frank Stahl, designed a test that could once and for all tell these models apart and give a clear answer. Their approach was just brilliant. They used 2 different versions, or isotopes, of nitrogen, a heavy version and the normal, lighter one. Now, since nitrogen is a key ingredient in DNA, they could basically create heavy DNA and light DNA. They started by growing bacteria in the heavy stuff until all its DNA was heavy. Then they switched the bacteria to a light nitrogen environment and just watched to see what happened as the DNA copied itself. This let them track exactly where the original heavy strands ended up. Okay, so let's check out the results. Here in Generation 0, this is before any copying has happened in the light stuff. All the DNA is tagged with that heavy nitrogen. When they spin it in a centrifuge, it all sinks and forms one single dense band at the bottom. This is our starting line. Now this is the crucial first clue. After just 1 round of copying in the light nitrogen food, that original heavy band is completely gone, and in its place, a single new band floating right in the middle, an intermediate density. This one result immediately kills the conservative model. See, that model predicted 2 separate bands, one heavy and one light. So, boom, our first suspect is eliminated. So then they let the bacteria copy themselves 1 more time. The final piece of the puzzle appeared. They saw 2 distinct bands, one still at that intermediate level and a new one up at the light position. This result was the nail in the coffin for the dispersive model, which would have just predicted a single blended band that just kept getting lighter and lighter. The case was just about closed. And this is just a perfect example of the power of a really good experiment. The results, that intermediate band in generation 1, followed by an intermediate and a light band in generation 2, that was the exact fingerprint that the semi-conservative model had predicted all along. Case closed.
All right, so now we know what happens. Each strand serves as a template for a new one. The next obvious question is, how? This isn't magic, it's the work of a super coordinated team of molecular machines, a whole set of enzymes that carry out this whole process with just breathtaking efficiency. The best way to think about it is like a construction crew. You've got helicase, that's the unzipper, it unwinds the double helix. Then DNA primase comes along, the initiator, and it lays down a short little primer to mark the spot to start building. The star of the show is DNA polymerase III, the master builder, which synthesizes the new DNA strand. After it's done, polymerase I, the cleaner, comes through and replaces those primers with proper DNA. And finally, DNA ligase, the gluer, seals everything up nice and tight.
And the main work site for this whole crew is called the replication fork. It's this Y-shaped junction where the parent DNA is actively being unwound, creating two single-stranded templates for the polymerase enzymes to get in there and read. And this fork is just constantly moving down the DNA molecule as it gets copied.
Okay, now here is where things get really, really clever. There's a big problem. The two DNA strands are what we call anti-parallel. They're like a highway with lanes going in opposite directions. But our master builder, DNA polymerase, is a one-way vehicle. It can only build in one direction, from what's called the 5' to the 3' end. So how on earth does the cell copy both strands at the same time?
Well, for one of the strands, it's actually super easy. This is the leading strand. The polymerase can just hop on and cruise along right behind the replication fork as it unzips, making a new, continuous strand of DNA without any breaks. It's a smooth, straightforward ride. But the other strand, the lagging strand, is pointed in the wrong direction. So the polymerase has to get creative. It has to work backwards, away from the moving fork. To pull this off, it synthesizes the new DNA in short, little discontinuous bursts. It's kind of like having to build a road by paving small sections, but in reverse. These short little sections of DNA that get made on the lagging strand are called Okazaki fragments, named after the scientists who discovered them. Each fragment is only a few hundred or maybe a couple thousand building blocks long, and the cell's job is to eventually stitch them all together into one seamless strand. So let's break down that stitching process. First, Primase lays down a little RNA primer to kick off the new fragment. Then DNA polymerase III zips along and synthesizes DNA until it bumps into the primer from the previous fragment. Next, the cleanup crew, DNA polymerase I, comes in, yanks out that old RNA primer and replaces it with DNA. And finally, DNA ligase comes and seals the final tiny gap. And this happens over and over and over again for the entire length of the strand. So far, we've mostly been talking about this process in simpler things like bacteria with circular DNA. But eukaryotes, that's organisms like us with long linear chromosomes, we face some pretty unique challenges, especially when it comes to speed and, well, what to do with the very ends of the DNA. I mean, to copy our massive genomes in a reasonable amount of time, we can't just start at one point. So eukaryotic chromosomes have thousands of places where replication can start. It kicks off at all these points at the same time, creating these replication bubbles that expand and eventually merge together. Each of these bubbles is called a replicon, and this kind of parallel processing is what makes it possible to copy billions of base pairs in just a few hours. But there's one final, critical problem with having linear chromosomes. On that lagging strand, once the very last RNA primer at the absolute tip of the chromosome gets removed, there's no way for DNA polymerase to fill that gap. This means that with every single round of replication, the chromosome would get a little bit shorter. Over a lifetime, that would be totally catastrophic. But, of course, nature has a solution. It's an enzyme called telomerase. It adds these protective caps, called telomeres, to the ends of our chromosomes. You can think of them like the little plastic tips on a shoelace that keep it from fraying. Telomerase actually carries its own little template and uses it to extend the end of the chromosome. This extension gives the regular replication machinery just enough runway to finish the job, preventing any important genetic info from getting lost.
And this brings us right to the cutting edge of science. Telomerase activity is very closely linked to how our cells age. In fact, most of our normal body cells have it turned off. But cancer cells, they often find ways to turn it back on, which basically gives them a kind of immortality. So understanding and maybe one day controlling this fundamental process of replication, it isn't just a biological curiosity. It really holds the keys to some of the biggest challenges in human health. The story is definitely not over.